Hello all, welcome to a new session of biology classes with Lakshmi Chandra. We were covering the various topics under molecular basis of inheritance, in which we were learning about that wonderful molecule called as DNA. In today's session, I will take you through a journey of a question. The question is, why we all are looking so different, yet we have some similarity, isn't it? We are similar to our parents, but not exactly the same, but we have some similarity. So this similarity and differences among us, all these things are the play of a particular segment of DNA. And that segment of DNA is called as a gene. So G-E-N-E, a gene. So gene is a part of DNA that is bringing in all these differences and similarities among us. So let's today learn about what this gene is, how it is functioning, and how it is bringing in all these similarities and differences. And of course, the whole story behind that, I cannot cover this in just one session. So the whole story of how a gene is actually bringing in these differences and similarities will be dealt with in a series of sessions from now. So today, let's understand the first process behind that. So that is transcription. So our topic for today is transcription in eukaryotes. Transcription in eukaryotes. That is how this gene is working, the first stage of how this gene is working. So I am keeping on telling about this term gene every time. So let me just tell you in short what a gene is. So I am writing here gene. And as told, gene is a part of DNA. It is a segment of DNA. So let me just draw a segment of DNA here. And in this, I am taking this particular part of this DNA. So this, I am calling it as a gene. So why do I call this as a gene? And the rest of the part here, say if I extend like this here, or if I extend like this here, the part here, this I am not calling that as a gene. But whatever is enclosed here within this particular area, I am calling it as a gene. So I can say that gene is that segment of DNA which is functional. Which is functional. We have 23 pairs of chromosomes, and chromosomes are made up of two components, DNA and proteins. Isn't it? So among that, the whole DNA that is present in this 23 pairs of chromosomes, the whole part of the DNA is not functional. Only certain segments are functioning. And those segments which are functional, you call them as gene. Now when I say my hand is functioning, my eyes is functioning, or my ears are functioning, we can understand that we are able to do something with those organs. But when I say a DNA segment is functioning, what does that mean? That means functional segment of DNA, I called it as a gene, isn't it? Now what is the meaning of here, the functioning of DNA? What is that? So that we have to understand in order to understand what a gene is. So basically, functioning means the DNA is having proper information to produce a protein, or to produce another molecule, or to pass information. So if the segment of DNA is having the exact information to produce a protein or to pass any information into the cell, then you call that part of the DNA is functioning. If certain segments of DNA don't have this information with them, you say that is not functioning. Okay, so the functioning of parts of DNA means it is having information to produce a product. So here in transcription, what is the DNA producing? The DNA is going to produce another nucleic acid. That's right, mRNA or ribonucleic acid. Deoxyribonucleic acid, DNA, is going to produce another nucleic acid, that is ribonucleic acid. So those segments of DNA which is having the information to produce RNA, you can call them as genes. Now RNA is not a simple molecule. DNA is only just one type of DNA we have in our cells, but RNA is not so. There are many different kinds of RNA molecules available. Okay, and it is all produced by this DNA only. But there is a specific kind of RNA which is carrying information to produce a protein, that you call it as a messenger RNA. So yeah, we'll deal with the different kinds of RNA in this session itself. So let me just make clear about this gene. So as I told, gene is the functional segment of DNA. So functional segment meaning also you might have understood. Now I am dividing this whole functional segment of DNA, that is gene, again into two parts. Okay, one part is this one, another part is this one, and in between there is a part. Okay, so I can say this is 1, this is segment 2, this is 3, this is 4, and of course this is a small segment here, 5. So this gene, I have divided them like this. Now why? Among these, the segment 2 and segment 4 is having the information to produce a protein. Segment 4 is also having the information to produce a protein. But segment 1, 3 and 5, of course they are functional, they are producing RNA, but they don't have the information to produce a protein. So among gene, there are regions which has the information to produce a protein, and there are regions which don't have this information to produce a protein. So the gene is split here. This is a special characteristic of eukaryote. Without knowing this, we will not be able to understand what is transcription and how this is done. Okay, so segment of gene which is producing a protein and which is not producing a protein. So again they have got special names. So those segments of gene which is able to produce a protein, you call them as the exons. So segment number 2 is an exon. Again segment number 4 is also an exon. Now there are regions in between them which is having the information to produce RNA, but it cannot produce a protein. So what do you call them as? They are in between these exons, or they are intervening the exons. So those sequences are called as introns. So segment number 3 is an intron, segment 1 is an intron, segment 5 is also an intron. So this kind of gene where you have both introns and exons present, you call this type of gene as a split gene. And this kind of split gene is not present in prokaryotes. That's why in the previous session where I had taken regarding transcription in prokaryotes, which was just taken before this session, there I didn't mention about these terms because it is not present in prokaryotes. But here in eukaryotes, before going straight away into the topic of transcription, we should clearly understand the difference between what is gene, and in that gene, what do you mean by intron and exons. So I hope this much is clear to you. Now moving ahead. How are these DNA molecules arranged in a eukaryote? So when I say the word eukaryote, that means true karyote or true nucleus or cell having a membrane bound nucleus. So our DNA is not present simply anywhere in the cell, but it is there present in a special place called as nucleus, and that nucleus is having a boundary also, a membrane also. And how is the DNA packaged? Hope you remember my previous session of DNA packaging. Okay, if you want to understand clear regarding that what is DNA packaging, how it is packaged and kept inside the very small nucleus. The 2-meter long DNA is coiled and kept into a very small nucleus, the microscopic nucleus. How that is were dealt with in the previous sessions. So in order to know details about that, please subscribe the channel and go through the playlists that you can just take one after the other. It is kept in proper order as it is there in your NCERT textbook. So it will be very easy for you to refer and understand. Okay, so coming back, our DNA is packaged inside the nucleus with the help of histone octamer. Histone is a protein. There are 8 histone proteins around which the DNA is wrapped and kept. In certain areas of the chromosome, DNA is very tightly wrapped and kept. In certain other areas, it is loosely wrapped and kept. So according to whether it is tightly wrapped or loosely wrapped, you have 2 types of areas in the chromosome. Okay, and those areas you call it as heterochromatin and euchromatin. Euchromatin is true chromatin or active chromatin. That means they are loosely bound and kept so that they can, it is very easy for them to act. But it's not with the case of heterochromatin. Heterochromatin is very tightly bound and kept. This DNA is very tightly bound over the histone proteins, and the histone proteins are very closely kept together. Okay, and so what happens? They are not able to function as a gene properly. Even though they have the information, they are not able to function as a gene. So what we have to do first in transcription or in the process of carrying the information from DNA to RNA, that is transcription. Carrying information from DNA to RNA or synthesis of a new RNA molecule. Okay, for that what has to be done? This tightly bound parts of the DNA, if it has the information to produce the RNA, that tightness, it has to be removed. It has to be made a bit loosened. That means heterochromatin has to be converted to euchromatin. So that step is the first step in transcription, and that is called as activation. Activation of the chromosomes. So activation of the chromosome here means if there are the segments, if genes are present in heterochromatin, these heterochromatin are very tightly packed, so they cannot get activated on their own. So they have to be converted to loosely packed structures, and that is called as euchromatin. So only then they get activated. If it is euchromatin present, straight away we can go to the next step. But if it is a heterochromatin, we have to go for, or the cell has to go for activating it. Now how this is done? Activation. So there are 2 steps basically for activation. One is acetylation, it's a reaction. Another one is activation marks on histones. So acetylation and activation marks on histones. It's very simple. Heterochromatin is converted to euchromatin with the help of an enzyme called as HAT. HAT. Now what is that HAT? Histone acetylation transferase. Histone acetyl transferase. Okay, HAT. Let me just rub out this because I hope you understood regarding what gene and intron and exons are. So the first step in activation is acetylation, and that acetylation is done by the enzyme called as HAT. HAT stands for histone acetyl transferase. This enzyme helps in converting heterochromatin to euchromatin, but this enzyme is not alone. It makes use of lots and lots of other proteins also. So they together convert heterochromatin to euchromatin. So now they are activated, and not only that, if that chromosome is completely activated or if that chromosome is ready for transcription, then there are some signals, there are some activation markers present on the histone molecules also. So which are those activation markers? One is ubiquitination, that is, you can see here UB on H2B. I have written here H2B, H3, H4, etc. They represent different histone molecules, okay, in that histone octamer. So the first molecule H2B, if ubiquitin is there on it, that means yes, it is ready. Next one, H3 has to be acetylated. Acetyl compound should be present on H3, only then it will be ready. Again on H3, another important thing is there, that is methylation. Acetylation followed by methylation, where in the lysine residue of histone. You know, histone is a positively charged protein and it is rich in positively charged amino acids like arginine and lysine. So when you take the component H3 of histone, in the fourth lysine of this particular histone, it has to be methylated. Sometimes 2 methyl units are added, sometimes 3 methyl units are added into the lysine, which is coming as the fourth residue, fourth amino acid. Into that, methyl groups are added. So that means it is ready for transcription. Again, when you take histone 4, again histone 4 also has to be acetylated. Only then we can say that our DNA is now ready for transcription. So that means it is getting activated. It is now active and it is now ready for transcription. Now that our DNA is activated and it is ready for transcription, let's see who is the key player that is doing this whole process. So of course this is a complicated process and just one particular molecule cannot do the whole thing, but there is a key player or the captain of this particular process. So that captain, let me introduce, RNA polymerase. So this enzyme, RNA polymerase, is the key player in this whole process of transcription, but not alone. This is having so many other factors also which is aiding or helping this RNA polymerase to carry out this. Now see, from the word RNA polymerase, it is evident RNA is getting polymerized or RNA is getting formed, and yes, that is our process of transcription. So what is transcription? It is carrying information from DNA to RNA. There is some information in DNA that is passed on to RNA. So what is formed there? RNA is getting formed. So you can say that transcription is the transfer of information from DNA to RNA, or it is the formation of RNA based on a DNA template. And who is the key player here? It is the RNA polymerase. And already I have introduced regarding this particular enzyme in the previous session of transcription in prokaryotes. There we had learned that there is only one enzyme, RNA polymerase, which is carrying out the whole process of transcription. And all the different RNAs are produced from this one particular RNA polymerase enzyme. But in the case of eukaryotes, it's not so. Eukaryotes don't have just one RNA polymerase. Instead, they have 3 different RNA polymerases designated as RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. And each of them, they produce or they help in producing, synthesizing different RNA that is present in the cell. Now as you know, the 3 different major RNAs present in a cell are rRNA, where here small r stands for ribosome or ribosomal RNA. Another one is mRNA, and the third one is tRNA. mRNA, M is for messenger RNA, and tRNA, T is for transfer RNA. So these are the basic three very important RNA molecules present, and among this rRNA there are again different types, and they all vary in their S. What is this S here? Their densities, which is measured in a unit called as Swedberg's unit, and that Swedberg unit is denoted by the letter S. So based on that, there are different types of rRNAs. They are designated as 18S, 5.8S and 28S. For exam, you can easily learn it as 1 to 5S, 1 to 5, that is 18 is having 1, 2 and 5. Okay, and all of them ends with 8. Okay, and only in after 5 it is 0.8. So 1 to 5S, like that you can learn. 18, everything should end with an 8. 18S, 28S, 5.8S. So what is that S there? That is the measure of density in a unit called as Swedberg's unit. Okay. So the three different rRNAs are synthesized by RNA polymerase I. That's the key player. And where is this happening inside the nucleus? It is happening in a place called as nucleolus. You know about nucleolus, isn't it? A structure, a dense dark structure present inside the nucleus. So that is the location, that is a place where rRNA is getting synthesized. And who is doing this? Who is the key player there? It is RNA polymerase I. Now moving on to mRNA. This is synthesized by RNA polymerase II. But keep it in mind, it is actually the precursor of mRNA which is getting synthesized by RNA polymerase II. It is in all cases this is the precursor. Precursor means prerequisite. That means it is formed in a raw form, and later on it is getting processed. Okay, it is getting modified to produce, to give you the correct mRNA. So we'll be learning about that. Don't worry on that portion. So here just understand RNA polymerase II is producing this mRNA, and also snRNA, SN in small letters, that is small nuclear RNA, SN, small nuclear RNA. And this is happening inside the nucleoplasm. Again, RNA polymerase III is involved in the formation of tRNA and also 5S ribosomal RNA. Remember, there is no 8 here. Okay, so all other rRNAs are here, ending with 8 that I have told you. So likewise, you can remember for your exam also. You will not have any confusion. So RNA polymerase III is involved in the production of tRNA, 5S rRNA. Okay, 5S rRNA, and this also takes place in the nucleoplasm. So if you can just register this particular tabular column in your mind, it will be easy for you to write for your exam and score marks, and also for multiple choice questions, location or which RNA polymerase is synthesizing which one, and all these things you can get it very easily from this tabular column. So in today's session, I just cannot take about how transcription of all these RNAs are taking place. Okay, so I'll concentrate on RNA polymerase II and their activities, or how mRNA precursor is getting synthesized. So when I say transcription, I have to say about all the RNAs, how they are getting synthesized, because it is carrying information from DNA to RNA. In today's topic, we'll study about how information is carried from DNA to mRNA or messenger RNA. Okay, and in that messenger RNA, there are various steps involved in the process of producing messenger RNA from DNA. From the word itself, it is there, messenger RNA. It is carrying a message from DNA. It is having that message, and it will deliver the message elsewhere in the cell for the production of another protein. So that particular series, that is what I have started, when I started the session I told you, why there is so difference and similarities. It is all because of this message that is carried from DNA. Because of the differences in these messages, this whole process happens. So let's understand about this key player, this key part, messenger RNA. Okay? And the whole process of transcription of messenger RNA, you can divide it into 4 different steps. That is initiation, elongation, termination and processing. Initiation, the starting, then the process proceeds forward, elongation, finally stops, termination, and then there happens some modifications to produce the finished final mRNA. Okay, so let's see one by one. Let's see what is happening in initiation. In initiation, first step is identification of promoter, followed by interaction of RNA polymerase with the promoter, and then promoter clearance. So here I am keeping on saying the word promoter, promoter, promoter. So first I must tell you what a promoter is. So moving on to promoter. In order to understand what a promoter is for transcription, let me first tell you something regarding unit of transcription. So as told earlier, the whole DNA is not coding for anything, that is not functional, but there are certain regions which are functioning. Okay, so there are certain regions which are active transcriptionally. So those regions which are active transcriptionally are called as the unit of transcription. So here imagine if this represent a DNA. Among this I have demarcated a region here, and this segment is the unit of transcription. Now let me tell you which are the key players or which are the key segments of this unit of transcription. So look at the second diagram that is shown here. So this second diagram is showing this particular part, that is this unit of transcription is expanded and shown here. If you just look carefully, you can just understand there is a point here called as the start point. So this is the point where the whole process of transcription begins. And that start point is labeled here as +1. Like in mathematics, there is a number line, no? There is 0 point, and towards the left side, we put all the negative numbers, towards the right side, we put all the positive numbers. Likewise, if you just imagine in a transcription unit like this, the start point you take it as +1. There is no 0 over here. The start point you take it as +1, and towards the left side that you go, you call that area as the upstream. And you keep on marking here the numbers -1, -2, -3 likewise, and ultimately you come to an area called as -25 region. And again, if you still go if you move for upstream again, you will come to an area called as -80 area. These two areas are very important areas in the case of transcription in eukaryotes, and those regions, they actually tell the promoter sequence. So they are the regions which are pointing towards the promoter sequence of eukaryotic transcription. So here again, from the start point, if you move downstream towards the other end, that is called as the downstream. If you move, so you will be marking there as the numbers +2, +3, +4. So positive signs towards the downstream, and towards the downstream is actually present our gene. That is the functional part. So what is this promoter doing? It is the regulatory part. Okay, then comes the functional part, and as told, the eukaryotic gene is a split gene having introns and exons. So that is what is shown over here. And ultimately this comes to another region called as terminator, where there are signals for halting or stopping the process of transcription. Okay, so the unit of transcription has basically three regions. That is the promoter region, then you have the gene, so specifically you call them as the structural gene, followed by terminator. So these are the three units or three segments or three parts of unit of transcription. So what is this unit of transcription? It is that segment of DNA where the whole process of transcription occurs. And as I told you, minus 25 and minus 80 region, which is part of the promoter region, it is very important, I told you. If you just carefully examine here, in the minus 25 region, you can see a sequence, consensus sequence it is called as. It is commonly occurring in almost all eukaryotic organisms. Towards the minus 25 region, you can see the sequence TATAA sequence. So that is called as the, commonly it is called as the TATAAT box. TATAAT box. And again, if you go upstream, you call this area as upstream, you can go upstream, you can see another sequence CC, it will be followed by here C likewise. So but this area is the important area, CAAT region. Okay, so this is called as the cat box, CAAT. So as I told you here, the minus 25 and minus 80 sequences are very important. They are the promoter sequences, and in that promoter sequences, specifically the promoter sequence is marked by TATAAT box. TATAAT box will be present, and in majority of the cases, cat box also will be present. Again, if you go downstream, suppose minus 100, there will be other such consensus sequence also present, but these two are commonly asked and most important consensus sequences. So they form the promoter. Now, as I told you, cat box and TATAAT box are important because they form the promoter. There is one more importance of this particular sequence. See, here you can see that I have written the CAAT sequence or TATAA sequence on one strand of this particular segment. That is, I have written this on the strand that is going from 5' to 3' direction. Okay, so the other strand I have not written here. So here whatever sequence is coming, it will be complementary to the TATAAT box or the cat box. That is obvious. Now this strand which is having the TATAAT box or the cat box, okay, the sequences in that strand will not synthesize RNA, but the sequences which is complementary to that strand will synthesize RNA. That has the information to synthesize RNA. So which one among this will synthesize RNA? It is not the strand which is containing the TATAAT box or the cat box, it is the other strand. So it is the strand which is running here from 3' to 5' direction that is acting as the template strand. So the TATAAT box or the cat box also tells you which is the template strand, the strand just opposite to that. So then what will you call this strand as? This strand is called as the coding strand. It doesn't code for mRNA, but its sequences, its codes will be exactly like the mRNA. Just the only difference is instead of thymine, uracil will be present in mRNA. That's the only difference. So, which is the template strand? The strand opposite to the one having TATAAT box and cat box. So, these two tells actually which one is the template strand and which one is the coding strand. Now imagine this TATAAT box is in the other strand. This is 5', I am writing here 3', okay, and 3', and here imagine cat box is here, TATAAT box will be here, okay. Then if this is the start sequence, then which strand will be the template strand? This is going to be the template strand, isn't it? Then where will be the structural genes? It will be towards this direction. Okay, so in this case we are taking this strand. So structural gene is towards this direction. Here you can see structural gene is towards this direction. Okay, so always remember that TATAAT box and CAT box are utilized to understand or to demarcate two things. One is which is the promoter site. Another thing is which strand is going to act as the template strand. So those two things. So here we understood which one is the template strand and which one is the coding strand. Now again, coming back to this particular structure, it is understood that this strand running from 3' to 5' here is the template strand, and this region is the promoter region. Here you have the gene, the structural gene. So this structural gene will produce an mRNA, and this mRNA in the case of eukaryotes will produce only one protein. So that is again coming to another difference. Okay. In the case of transcription in eukaryotes, one mRNA is going to produce only one particular type of protein. So such kind of a gene, you call it as a monocistronic gene. The gene is producing just one mRNA, and that one mRNA is producing only one kind of protein. You call that kind of a gene as a monocistronic gene, and that is a speciality of eukaryotic cell. One gene, one mRNA, only one protein. Now in the case of prokaryotes, this particular segment of DNA can produce more than one type of mRNA, and that will keep on producing more than one type of protein, which all together will function into carrying out, say, one particular biochemical pathway. So more than one proteins are produced. So you call such kind of genes as polycistronic. Okay, so monocistronic and polycistronic. So now what a promoter is. When I tell you what a promoter is, immediately what will come to your mind? TATAAT box and CAT box. Now since we have understood what a promoter is, let's now enter into the first step of transcription, that is initiation. Understood what a promoter is, let's move on to the process of initiation. So our DNA got activated, we understood what a promoter is, okay, and let us see how this initiation is taking place. So in this initiation part, I have already introduced to you regarding what is TATAAT box, that is in -25 region, and this part you call it as the core promoter region. Again, if you go further downstream, there is another box called as CAT box towards -80 region, and again further down if you go, there is a -100 region which is rich in GC sequences, and you call this as a GC box. So here the CAT box and the GC box together forms another part of the promoter called as the regulatory promoter. This regulatory promoter is also there down. Again, if you go, I have just bent and I have drawn here. Again, if you go down, that means if you go keep on going upstream towards the -1000 base pairs, approximately -1000 base pairs, you can see a region termed as the enhancers. Okay, so all these are part of our promoter only. So basically the core promoter, the main promoter region is this region, -25 region, where you have the TATAAT box. Now why that is called as a core promoter region is because that is the area where, as I told you, the key player, that is RNA polymerase, comes and binds. Okay, so RNA polymerase is having a great affinity for DNA as such. It is having a great affinity for DNA. Wherever it sees the DNA molecule, it goes and binds. But for transcription to begin, if this RNA molecule is going and binding anywhere in this DNA, that is of no use. It has to specifically bind itself towards that core promoter region. That is the region near the TATAAT box. It has to go and bind. So now preparations are for that. Preparations are to make this TATAAT box ready for this RNA polymerase to come and bind. That is, now we are going to activate the TATAAT box. How? For that, we have these 4 players to activate the TATAAT box. The core promoter, they are the transcription activators, you call them as, architectural regulators, co-activators, and general transcription factors. General transcription factor 2 it is written. In short, it is written as GTF2, or sometimes it is also called as TF2, transcription factor 2. This 2 stands for because it is going to help RNA polymerase II to do these activities, because we are dealing with mRNA which is synthesized by RNA polymerase II, as I told you earlier in the tabular column. Okay, so that is why it is written here GTF2, general transcription factors 2. So this transcription activators, which is written here 1, they will go and bind to the enhancers. So I'm just putting here 1 here because to show this one. Transcription activators, they go and bind to the enhancers. So if the transcription activators, imagine transcription activators are here, they have bound themselves with the enhancers. Now this binding of transcription activators with the enhancers sends a signal to this GC box, CAT box and all to activate the near TATAAT box, which is sitting near to that. Okay, so transcription activators, they bind to the enhancers and indirectly they are activating the core promoter or the TATAAT box. Now next one is architectural regulators. These are all proteins. Okay, these architectural regulator proteins, they come and bind here. I am putting here number 2 to show this. These architectural regulators, let me write here AR in short. Okay, these are another kind of proteins which go and bind to the DNA and they try to bend the DNA in a manner so that now again these enhancers are in close proximity with the TATAAT box. Okay, so first step, transcription activators, they go and bind to the enhancers. Next, this enhancer should come in close proximity with the core promoter, that is with the TATAAT box. For that, the DNA is first just bent in a manner. For that bending, that is to change the architecture of the DNA, you have the protein architectural regulators going and binding to the DNA and bending it. Now what happens? The enhancers are now almost near to the TATAAT region. Now another protein, that is the co-activators or the mediators, they come and bind over this one. Here the co-activators or the mediators, they come and bind. When these co-activators come and bind to these enhancers, it will send a signal directly to the TATAAT box. Yes, now you should get ready for transcription. Soon RNA polymerase will come to transcribe. Okay, so all these are steps that is actually making this TATAAT box ready for this transcription or for this RNA polymerase to come and bind. Like for example, when you go for marriages and all, we can see the bride and the bridegroom, they're getting ready to meet each other, isn't it? Same way here we have the RNA polymerase II and the TATAAT box. RNA polymerase II should go and bind with the promoter. For that, first the core promoter region is getting activated. It is making itself ready for that. And for that, who all are helping them? Transcription activators binding to the enhancer. Again, there is a change in the structure, architectural regulators, followed by the co-activators which act as a mediator. This co-activator is actually acting here as a mediator between the enhancer and the core promoter. And of course, there are key players called as general transcription factors. There are many general transcription factors which go and bring the RNA polymerase. They recruit the RNA polymerase towards this particular promoter region. Okay, so here I have written in nutshell, that is enhancers plus GC box plus CAT box, they are all regulatory promoters. They all help in enhancing, in activating the core promoter. Core promoter is basically in the -25 region, TATAAT box, and the RNA polymerase II attaches to, which among these two? RNA polymerase attaches to the core promoter. That is what is called as core promoter, after the activation of the regulatory promoter. So you have in the promoter two promoters are there, regulatory and core. The regulatory promoters have to be activated first, which in turn goes and activates the core promoter. Once the core promoter is activated, RNA polymerase II can go and bind. Okay, now how the regulatory promoter is activated, we have already told you. Now, how this RNA polymerase is going and binding? So, for that it needs general transcription factors. So, let me just introduce to you the major general transcription factors. Now, once this core promoter region is getting activated, the initiation begins. So, the first step, initiation, activation of core promoter, that we have already learned. Next is formation of a pre-initiation closed complex, followed by pre-initiation open complex, and then promoter clearance. So once we learn these steps in initiation, one after the other, it will be very easy for us to understand how this process of initiation of transcription is taking place. Okay, so just understand each process as if you are seeing a film. Okay, there are some key players over here, there are some characters over here, and each character has got their own role here to play in this process of transcription. So let's first understand who are those characters. These characters are here the transcription factors. I have told you the general transcription factors. So those general transcription factors. So let us just see who all are they, the general transcription factors. They are denoted by different alphabets. Transcription factor 2D. So transcription factor 2 means it is helping the RNA polymerase II, and first one is transcription factor 2D, which needs the help of TBP, that is TATAAT Box binding protein. Okay, it always needs the help of this one. Next one, TFII A, followed by TFII B, and then F, E, and lastly the H. So this is the order, DA, BF, EH. Now how to remember this order, one after the other, which one is coming and doing? Let's just understand, like a girl named Diya, okay, had an apple, A, TFII A, and then she started playing with a ball. She got tired and she's going to sit under a fan. Suddenly she sees an elephant is coming. Okay, elephant is coming, and then she looks up and she sees a helicopter flying. So likewise you can remember, Diya ate an apple, playing with a ball, got tired and sitting under a fan, F, sees an elephant, okay, and she looks up and finally she sees a helicopter. So if you just remember this particular sentence that I've told you, you'll just understand which TF factor, transcription factor, is coming one after the other in the process of transcription and doing what kind of function here. Let's focus on that. So now our attention is here in the core promoter, that is in the TATAAT box. As I told you before, the TATAAT box got activated, the core promoter got activated. Now we have to see the formation of closed complex. So for that, the first transcription factor is TFII D. Now why all these transcription factors are there? It is needed for the RNA polymerase to come and bind to the promoter, core promoter. Okay, promoter got activated. Now who has to come? Promoter is waiting for whom? It is waiting for the RNA polymerase. Now can RNA polymerase come simply like that? No, it also needs to be carried. It should be recruited by someone, recruited. So for that, again processes are happening. For that only these transcription factors are functioning here, right? So first transcription factor 2D along with TATAAT binding protein. So this TATAAT box binding protein and this transcription factor 2D. Okay, so this I am marking it as transcription factor 2D, and this is having along with that, what is there? TATAAT box binding protein. It is first coming and attaching to the TATAAT box. Because it has the TATAAT box binding protein, the TATAAT box binding protein TBB will directly take this TF2D exactly to the TATAAT box to allow it to fix with the TATAAT box. And you see this TBP, it attaches to the TATAAT box like a clamp. Okay, and then bends this DNA around 80-degree bends. So the DNA is like this, and once this is getting attached, okay, our DNA slowly gets bent like this. Okay, I hope you're following, and this is because the TBP, the TATAAT box binding protein, is acting like a clamp, and our transcription factor 2D is sitting over this one, and it is just applying the force, and this is getting bent. Now this bent shape will allow the other transcription factors to come and join. So who is the next one to come and join? Dia ate an apple. So it is transcription factor A which should come and join. Okay. So transcription factor A, I'm just putting here A. It is joining along with this one, and the main function of this is to strengthen the attachment of the TF2D over this particular DNA or over the promoter. It strengthens or it stabilizes the attachment. That is the function of TF2A. Now Dia ate an apple, and then what she started doing? Playing with a ball. So next factor is TF2B. Okay, now TF2B, what it does is, it will stimulate the TF2F to bind with RNA polymerase II. Now TF2B is coming and binding. Let me put it here in this area. TF2B is coming and binding here. As soon as TF2B is coming and binding, what happens is, so this head and tail that I have drawn is our key player, that is our RNA polymerase. Our RNA polymerase II. This tail also have got a role, okay, that it has got a great role that I'll tell you later. So here RNA polymerase is there, and as soon as TF2B binds here, what happens? It stimulates the TF2F to bind with RNA polymerase II. So TF2F now comes and binds with RNA polymerase II. Once this TF2F is there with RNA polymerase II, this TF2F will slowly carry RNA polymerase, and it will allow the binding of RNA polymerase in that core promoter. Now our RNA polymerase, its tail here, along with the transcription factor 2F is there here in the promoter region. Yes, now came RNA polymerase is there in the promoter region. For this we were doing all these things, activation, and then one each factors were coming and binding, and it was making a proper area, a proper shape and everything for the RNA polymerase to come and bind exactly to the promoter region. And finally RNA polymerase have come here. Now again, to maintain the stability of this binding, transcription factor 2E comes and binds, so that the RNA polymerase, which is here, Okay, who is recruiting it? Factor F is recruiting it towards the region, and it is stabilized by factor E. So saw an elephant. So that's it. Okay, so if you just want to remember the order, Diya ate an apple, played with a ball, then was tired sitting under a fan, and then saw an elephant. So till elephant, till that E factor we have completed. Now comes another, the last factor, transcription factor 2H. This is a very important factor. Okay, so after the attachment till TFII, this complex that is formed here, you call this as a closed complex. Because till now the DNA strand did not separate, it did not melt away, it did not unwind, it is still together. So this is a closed complex. Okay, so pre-initiation closed complex is over. Now as soon as the factor TF2H enters into the site, this closed complex becomes open, because this TF2H is having a helicase activity. It can unwind the helix. Then it also has a kinase activity. What is the enzyme kinase doing? It adds phosphate, isn't it? That is phosphorylation. That property is also there with TF2H. Moreover, it also helps in nucleotide excision repair. So after the TF2E, finally the TF2H comes and binds. Let me write it here. Once the TF2H comes and binds, there occurs the formation of open complex. Because of the helicase activity of TF2H, the area, the two strands of the DNA unwinds. Okay, so how will the structure be now? It was an open complex, it was a closed complex, and now the area I can just draw like this. The two strands have now unwound and it is a open area. So here we have this whole complex. Once H is attached, here you have the TATAAT box, here you have the +1 site, okay, area just after this, here the two strands, the double bonds, the hydrogen bonds here is now unwound, okay, and these two strands separate out, they are separated, and this forms the open complex. So from closed complex it becomes open complex by the attachment of TF2H, because it is having a helicase activity. Now once the two strands are opened, okay, now our RNA polymerase has to move forward and forward. So it has to move forward and forward. Only then it can transcribe the whole strand of DNA. But you see here, the RNA polymerase is surrounded by these many factors. And all these factors are holding the RNA polymerase in the promoter region. But if those factors persist, RNA polymerase cannot move forward. So now RNA polymerase has to move away from the promoter, that is promoter clearance. Let's see on that. Promoter clearance. For that, let's look into the detailed structure of the tail of this RNA polymerase. As I've told you before, this tail has got some importance. Okay, this tail is often called as the carboxy terminal domain of RNA polymerase II, carboxy terminal domain or CTD. This has got a speciality. Okay, this has a heptad. Heptad means it is having 7 amino acid residues, and these 7 amino acid residues keeps on repeating in this order in its tail. Okay, see 1, 2, 3, 4, 5, 6, 7. Again after this, again this 7 keeps on repeating. So it's called as a heptad. And this heptad, the most important person in this heptad is the serine residue. There are serine residues in 3 positions of this heptad, position 2, 5 and 7. Among this, the serine residue which is there in position 5, this residue is getting phosphorylated. Phosphate is getting added. The residue number 7 everywhere in this heptad is getting phosphorylated. By whom? By the kinase activity of the TF2H. I've told you TF2H is having 2 activities. One, it is acting as a helicase, so it unwinds the helix, it has formed the open complex. Now the same TF2H is also helping in promoter clearance by adding phosphate into the 5th residue, 5th serine residue of this heptad of the carboxy terminal domain of RNA polymerase. So that is the first step, phosphorylation of serine residues at the 5th position. This is important. 5th position of a heptad by CDK7 subunit. So that is this unit I have specified, subunit. CDK7 subunit of TF2H. So TF2H is a key player here. Now, once the TF2H is phosphorylating, it is like giving a little bit fire to the tail of something. So what happens? Because of this fire, the RNA polymerase runs away from the promoter. It gets released itself from the promoter. All those complexes were catching hold of the RNA polymerase and making it sit comfortably in the promoter. Those transcription factors starting from TF2 D, TF2 A, B, everyone was just coming and stabilizing the presence of RNA polymerase II in the promoter region because that is needed. But once it is getting this phosphorylation in its tail, like fire in the tail, what happens? It tries to move away from the promoter and it goes downstream towards the structural genes. That is what we need also. Only once the RNA polymerase moves towards the structural gene, new mRNA will be produced. Okay, so once phosphorylation happens, what is the next step? Phosphorylation of the carboxy terminal domain, that is the tail region of RNA polymerase, acts as a trigger to separate the RNA polymerase II from that promoter complex. So in that promoter, now those factors are no more present except TF2D and the TATAAT binding protein. TF2D and TATAAT binding protein will still be present in that TATAAT box and it will be bent, the DNA will be bent and kept. Why? For other RNA polymerases to come and bind and to continue this process. Okay, so that will be left behind. TF2D and TBP will be left behind so that other RNA polymerases can come and bind to the promoter region easily and the same process continues. Okay, and so once the RNA polymerase enzyme moves downstream, now initiation is over and now we come to the next phase of transcription, that is elongation. Okay, so that elongation, again, it's a complex process like this. There are factors, elongation factors helping in elongation. So I don't think that I'll be able to complete that in this session. So please wait, I'll be uploading how the process of elongation is taking place in eukaryotes very soon in the next video. And my humble request to all my viewers to subscribe this channel so that you will get the different topics in your class 12 biology, which will be very useful for you if you are aiming at competitive exams, entrance exams. Okay, and these are some additional points which are not there in your text. So apart from your board exam, you can also use this channel for writing your other competitive exams in future. So wishing you all good luck. Thank you.